now have Dr. Hockett to introduce our next speaker.
It is my pleasure today to introduce Dr. Deutschbauer. So just a little bit of his background. He's actually originally from Pennsylvania. He did his BS at Carnegie Mellon University in 1998 in the Pittsburgh area. From there, he went on to a PhD at Stanford, followed by a short postdoc there before moving across the bay to Berkeley, where he postdoc for a few years, and then was hired at Lawrence Berkeley National Lab in 2008 and has been there since. In 2018, he also took on an adjunct appointment at UC Berkeley in the Department of Plant and Microbial Biology. And so he is an expert in bacterial functional genomics and systems biology. And for the past 10 years, his group is focused on developing and applying tools, genetic tools, to rapidly characterize previously uncharacterized genes in diverse bacteria, which we'll be hearing about today. And then I'll just wrap up with saying that his group has also been highly collaborative. So this was some of the work that my lab and Carolee Bull's lab, we reached out to him, and he was very gracious about helping us with some projects that a student of ours is working on. So if you see things that you like, definitely reach out. There could be a benefit to that. And then finally, we have a plaque for all of our speakers. So take it away.
Thanks, Kevin. Hopefully you can hear me on Zoom. Yeah, thanks for the invite to come here. This is really special. My wife's in the audience. She's a Penn State grad from years ago, and we started dating. She was an undergrad here at Penn State, so I spent a lot of time here at Penn State College when I was an undergraduate in Pittsburgh. So this is very special to be here. And I'm happy to give you sort of a high-level overview of the work that my lab does at Lawrence Berkeley National Laboratory. If you haven't been there, this is the view from the hill with LBNL, looking out over the bay. Golden Gate Bridge is somewhere over here, San Francisco. My lab is not up on the hill. It's moving up onto the hill to this beautiful location in about 1 year from now. My lab is actually at an offsite location, kind of where my cursor is right here, out in the lowlands of Berkeley. I take a pretty broad view of microbiology, and I'll frequently say that my goal is to accurately predict everything from a bacterium just from a genome sequence. So it could be a genome of an isolates or a metagenome assembled genome. And just given that information, that DNA sequence, I'd like to predict everything about that microbe. So ranging from more simple characteristics, like what substrates it eats, to more complex phenotypes, like regulatory metabolic networks. Then finally, what I think is the hardest thing to predict based on genome sequence is the actual ecological niche or activity of a microbe in its native context. This is hard for a couple reasons. The first is that my lab mostly studies non-model bacteria. So the majority of the microbes I investigate in my lab are things that we or my colleagues isolate. So the only thing that we have for them is the genome sequence after we sequence the genome. And there's EM images from my colleague Peter Whaley at LBNL, just showing images of some of the microbes that we study that are from the Oak Ridge National Laboratory Field Research Center. The second challenge is that there's a ton about gene function in microbiology and bacteria that we don't know. So it's probably not news to most people in the audience, but our ability to sequence genomes and sequence genes has far, far outpaced our ability to figure out what those genes and their encoded proteins do. There's this massive discrepancy. And with the rise of next-gen sequencing, there's been a massive explosion in the number of proteins in databases. This is from the UniProt knowledge base. It just shows the expansion of the entries in these databases over time. If you just focus on the green bar at the bottom, this is the SwissProt part of the UniProt knowledge base. And so the SwissProt part is the actual part that human biocurators look at the literature and convert that information into some kind of machine readable format in a database. It's a tiny, tiny fraction of what we sequenced. So less than 0.3% of all entries are reviewed by humans. And even of those, only a fraction have experimental evidence as to what the protein does. So I mean, our approach is quote unquote high throughput microbiology. It has to be high throughput because we study lots of different microbes. We constantly think about how we can miniaturize and multiplex assays to drive down cost. And I'm trained as a geneticist. So I make extensive use of genetic approaches to infer the functions of genes and pathways based on patterns of their mutant phenotypes. If you're familiar with DOE funding and how national labs work, it's very, very collaborative. The Department of Energy has a perspective that you get more from the sum of the parts than you would by funding individual groups to do research. I'm fortunate enough to be part of 5 big team-funded projects, 4 of which are funded by the DOE. We love acronyms. Enigma stands for something. MCAFE stands for something. So most of these are DOE funded. The ones with a star, I'll talk about research from those projects today. The only one that's not DOE funded is this GutWorks project. It's an NIH funded project. It's also a team project to do genetics and functional discovery of gut commensal bacteria. All right, so today I'll talk about our high throughput genetics pipeline that we've developed and optimized for the last 10 plus years. In particular, I'll talk about a technique called random barcode transposon-site sequencing, or RB-TnSeq, that we use for characterized gene function in bacterial isolates. I'll give the current status of this pipeline. I'll talk about 2 things that are like foremost on our mind right now. One is how we propagate our knowledge to new genomes, because we can't do experiments on every single organism. And the second is more about how we assign function in the first place. Currently, it's done manually with us staring at data, but that's not sustainable. And so I'll talk about some advances that could potentially be applied using machine learning. We'll then switch gears and talk about one group of bacteria in particular, so bacteria, the genus Rhodanobacter, and why we're interested in these, because they dominate at a metal-contaminated site in Tennessee. In particular, I'll talk about our findings from Rhodanobacter and how our success or lack thereof is leading us to develop some new genetic methods. One being genome-wide overexpression using bar-coded plasmid libraries. And the second that I'm really enthusiastic about is a direct genetic analysis of a microbe in the context of its community, which we call microbial community editing. All right. My lab makes extensive use of transposon-site sequencing. I didn't invent this approach. This is from a review describing the first 10 years of this approach, written by many of the labs that originally developed this approach and its variants. It's a very, very powerful approach. You can use a mobile genetic element, a transposon, to jump into the genome of a microbe. In this case, it lands in gene Y. It typically causes a loss of function of the gene that got mutated. You can make not just one mutant, you can make a whole bunch of mutants, right? You can make thousands of mutants. And you can then mix them all together into a library or pool. And the key insight is you can measure the importance of all the genes in the organism by competing all those mutants against each other under some selective condition that you're interested in. So this example, they're competing the mutants in a no antibiotic control, and then a second experiment where they treat it with an antibiotic. And by sequencing the population of the mutants before and after selection, you can get a measure of the importance of nearly every gene in the genome for fitness or growth in that particular condition. So I was telling Kevin before this, like when next-gen sequencing got invented, it kind of changed the entire way that we, at least I view, running these assays and opened up entirely new areas. I had done my PhD work in Saccharomyces cerevisiae, yeast, and there's lots of really awesome genetic tools that were developed in yeast. One of which was the yeast deletion library that had, in the place of a gene they deleted, they had DNA barcodes or tags that you could do these exact same kind of pooled fitness assays with, except you used a microarray to read out the strain abundance. And I knew that the power of that approach. And so, going from the inspiration of these TnSeq methods, we realized that biggest bottleneck in running these assays was the Illumina library prep cost. So we decided to incorporate the exact same DNA barcodes into our transposons that we had used in the yeast deletion library. And so the reason why this is really advantageous is that when we do one of our genome-wide assays, all we have to do is sequence the abundance of the barcodes before and after selection to measure the importance of all the genes. So we call this DNA barcode sequencing Bar-seq, and it's incredibly easy to do. The reason why it's so easy to do is you can put all of the Illumina adapter sequences directly onto the primers. So we can run thousands of these PCR reactions, mix the amplicons together, run it over a little Zymo column in 10 minutes. It's ready to be sequenced on the machine. There's no adapter ligation, DNA shearing, nothing like that. And to enable an economy of scale, we've put the exact same primer sites, U1, U2. These are the priming sites used for the yeast deletion collection uptag. We've put these into multiple different genetic systems. So we use the exact same priming sites and Bar-seq primers for RB-TnSeq that I'll talk about today, Overexpression, CRISPR-associated transposons. The three in blue I'll mention today. We also use it for lineage tracking and evolution studies, using transposon TN7 that goes into a neutral location. We use it for CRISPR interference. And I won't talk about it today, but we also use it for assessing genetic systems. And so we can barcode different constructs of plasmids to figure out which combination of promoters and drug resistance markers is ideal for a new target bacterium. Okay, if you can make a mutant library with this RB-TnSeq approach, it's straightforward to generate a big gene phenotype map. It's easy to run the library across many different experimental conditions. And so one organism that we've recently done this for is an obligate sulfate-reducing bacterium, D. sulfuribrio vulgaris. And it's from this study here. And so we've run hundreds of assays. And there's two high-level ways that we make insights about the functions of genes based on this data. The first is what we call a specific phenotype. So if a gene only has a phenotype in one or a handful of conditions, it's easier for us to assign a specific function to that gene. Versus if the gene is incredibly pleiotropic, it has phenotypes across hundreds of conditions, that tends to be harder. So in this example from D. vulgaris, this locus ID DVU2672 was a poorly characterized protein based on its computational annotation. The only time it's important for growth in one of these assays is when glycine is the only nitrogen source. So we've been able to figure out with additional experiments, this is actually a transporter for glycine. The second high-level way that we make insights based on this data is simply guilt by association, or what we call co-fitness. And so across all of the experiments, if two genes have the same pattern of phenotypes, they're likely to share functionality. This has been demonstrated by multiple groups. It's true for gene expression data, it's true for this mutant fitness data, it's true in any kind of case. In the case of the sulfate-reducing bacteria, we knew it could make all of its vitamins, but we didn't know what the genes were. It's kind of hard to see for making vitamin B6. And so one way to find the missing genes involved in making vitamin B6 is by asking which genes in the genome have a similar patterns of phenotype to a known gene in the pathway. So PDXA is a known gene in the pathway, and this uncharacterized gene DVU0826 has the same pattern of phenotypes across all of our experiments. And again, with additional experimentation, we were able to figure out this uncharacterized protein performed this step in the vitamin B6 pathway and sulfate-reducing bacteria. So this is a little bit older now, but I think it's valuable because it was, I think, the first good demonstration that we can take a non-just genome sequencing based approach to figuring out function of uncharacterized genes in bacteria, that we can take a more functional data-driven approach. And so we generated these RB-TnSeq libraries for 32 different bacteria and did around 5,000 of these genome-wide assays across those 32 bacteria. So this is about 20 million gene phenotype measurements. So one key thing is we're able to identify phenotypes for over 10,000 genes across these 32 bacteria that had poor or vague annotations. And in hundreds of cases, we could identify a very specific function for poorly annotated enzymes and transporters. And later on, when I talk about algorithms for machine learning to predict protein function, I'll come back to these annotated enzymes that we made based on genetics data. It's a lot of data. And like Kevin said, we collaborate with a lot of labs, both within our internal programs and with external collaborators. And so my colleague Morgan Price here is a guru in all things bioinformatics. And he developed this interactive website to explore all of the genetics data. And so there's lots of different functionality. My grad student Surya works on this NIH project. So he's working on doing genetics of gut commensal bacteria. And it's just an image of one of the genes, gene page from Foliola vulgaris. And it's a poorly characterized protein, has a vague annotation, can be dependent receptor. And you can see that that gene is important for fitness on raffinose and some related carbon substrates. Very often when we generate this data, one of the first things we want to find is whether or not somebody already published on this paper or published on this protein or a close homolog. There's nothing worse than like saying, well, we got this really novel insight. And then there was a JBAC paper published like 2 weeks ago about it. So the fitness browser has all of this RB-TnSeq data, but it's also linked to different computational biology tools. And one of them that I think is the best tool is this tool called PaperBlast. And so if you click on this little link for PaperBlast, it'll actually search through the published literature for papers that describe your protein and its homologs. And so in this case, only one hit comes up. There's only one paper that's been published that describes the homolog of this protein that I showed you. And it's a really distant homolog. So it's only 21% identity at the amino acid level. And if you look at the little snippet of text, it doesn't really say anything in detail about that homolog. So this is a novel finding, very exciting. Where are we now, right? National labs are always thinking about economy of scale and like doing big science. And so we want to scale this up even greater. We're nearly done making 150 of these mutant libraries now, including representatives of 100 different genera. So we've gotten better at library making. We buy a lot of chemicals. So we're expanding the number of conditions that we profile. So we define lots of different substrates and compounds and antibiotics from Sigma and other vendors. We have a really big focus right now on carbon usage. And I can maybe explain later if you're curious why that's the case. And the reason why this is all possible now is that the cost of sequencing keeps going lower and lower. So on the newest and greatest Illumina machine, this Illumina X10B flow cell, we can sequence over 3,000 of these genome-wide assays on one flow cell. So when we do one of these genome-wide assays, if you give me a sample to do Barseq on for an assay, it only costs me like $3.50 for the cost of sequencing. So I always say, Kevin mentioned, like, definitely reach out if you have a microbe that you want to try this on. And we're always really, really eager for some genera that we haven't done yet, but we're happy to do. A lot of people like pseudomonads. So the odds of you coming to me and saying, I have a pseudomonad, the odds are like reasonably high. People like pseudomonad. If you come with me with a gram-positive bacteria, traditionally, I would have sort of like waved my hands and said, that's too hard. But we have gotten a little bit better. So Anthony Shiver, who we work with in Casey Wong's lab at Stanford, has gotten Bifidobacterium to work. And I'll show you some results from that work in the subsequent slides. They're really exciting. Leslie Day was a visiting grad student in my lab from University of Minnesota, and she got this to work in Archaea. So she made libraries in 2 different methanogens, which is, you know, a really, really big accomplishment. We've also had success with MTB, with Sarah Stanley, UC Berkeley, Streptococcus, Rhodococcus. We've had more success recently. The less that thing for gram-positive bacteria, it's not a one-size-fits-all strategy for making mutants at a library scale in gram-positives. They typically require organism-specific delivery methods to make it work. Okay, but for Bifidobacterium, we're really excited about this because if you're, know the human microbiome field all that well, Bifidobacterium is really important in early human development. I wasn't involved in this work, but this paper kind of shows the progression of the microbiome in early human development. And this 3 to 6 month time point, this really big cluster here, this is fairly all Bifidobacterium. And this makes sense because Bifidobacterium are known to consume milk-derived oligosaccharides. This is from breastfeeding. They're associated with positive health benefits. But like all bacteria, there's gaps in our knowledge of how these organisms work in their pathways. So I like the example of Bifidobacterium because it sort of extends the sort of range of bacteria we do our genetics in. It's an important organism. And this study also kind of illustrates how we're operating now. And so Anthony did a real tour de force with this. And he made one of these pooled RB-TnSeq libraries. And we did our typical in vitro assays with different carbon sources across hundreds of different conditions. But of course, these microbes don't live in the well of a microplate. So Anthony also did assays in germ-free mice and chickens. Why he did chickens, I don't know. There was a collaborator in North Dakota who had a germ-free chicken facility. I'm not sure why, but we did chickens. And then I guess one downside of the pooled fitness assays, there's certain phenotypes in bacteria that doing the assays in a pooled format is not ideal. So genes that are secreted or secreted factors are not ideal to assay in a pooled format. So Anthony went through the efforts of rearraying the mutant library to a bunch of individual clonal strains that we can then put into the wells of a microplate. So we have a really big collection of clonal individual mutants that we know the identity of which mutant is in each well of the microplates. And so by assaying clonal mutants, you can look at things like cell shape or do metabolomic studies on them as well. So we did all of these things as part of the study. And I'll just walk through one slide showing you the findings that we have from running each of these 3 different kinds of assays. So Bifidobacterium, we look at its genome, it looks like it's missing a step in histidine biosynthesis. It grows in the absence of histidine, so it can make the amino acid. But if you look at the annotation of all its proteins, we couldn't find a step for histidinyl phosphate phosphatase. So like the example from vitamin B6 in D-cell Vibrio, we just compare fitness data in our in vitro assays in the absence of histidine to the presence of histidine. And we're able to quickly find this protein encoded by locus ID 0982 encoded the missing enzyme. Straightforward. The example in vivo in mice and chickens is not so clean cut and interpretable. So what you're looking at in these plots is the abundance of barcodes in the library represented by these particular mutants. And so we did these assays with a mouse standard diet, mouse fed a Western diet. I don't know what the MD diet is for mice. And we did the chickens using a standard chicken diet. And this just shows the progression in the abundance of these barcodes measured at different time points during the course of these colonization experiments in mice and chickens. So remember that the transposon mutant library has around 200,000 strains in it. So 200,000 different barcodes. And what's really striking is that after a week or even earlier, but certainly after 2 weeks, we don't see 200,000 barcodes anymore. We only see barcodes associated with a handful of mutants. Basically, mutations in these genes take over the population. in mice and chickens. And it's very, very, very strong, right? Basically, every single barcode that we see after 2 weeks in mice fed a Western diet are mutants in these 3 genes. What's also striking is that these are all different transport systems in Bifidobacterium. The one thing you quickly want to know is what substrate do these transporters transport? So here we can go back to the in vitro data, where we profile the population in the wells of microplates that different substrates. And we can see, for example, for this PTS system encoded by 1892 to 1894, that transporter in vitro was required for growth on glucose and fructose. So it's a transporter for simpler sugars. We're still parsing through this. Our best guess right now as to why we're seeing this phenomenon is that glucose is toxic to Bifidobacterium, which we're still trying to verify with mouse studies. We have some validation this is true if we do in vitro experiments with varying levels of amino acids. We're still trying to parse this out. The other thing I kind of throw my hand up in the air is that regulation in bacteria is confusing. And a lot of these transporters are connected to regulatory pathways. Anyways. And if you ever do assays with mice, this phenomenon of certain mutants taking over the population, it happens all the time. Not just for Bifidobacterium. It happens in all of the gut Bacteroides strains as well. All right, so the final example of assays that we run for Bifidobacterium are these assays that we can do with single isolated mutants that are not amenable to assaying in a pooled format. And so Casey Wong's lab at Stanford has developed a pretty slick system for doing high throughput imaging of lots of mutants using microplates. This is showing representative images of what the cells look like for different mutants shown above. So I didn't realize this going into this, but the Bifidobacterium is named for this bifid cell shape. So it has this like Y shape. If you look under the microscope of any Bifidobacterium culture, only a fraction of the cells will have this shape. It's a heterogeneous population. Some fraction look like bifid with the Y shape, and others look more, I guess, bacilli-like, not the shape. And so one outstanding question are what are the genes and pathways that drive an increased fraction of this bifid cell shape formation? And so we imaged all the individual mutants that we counted, what fraction of the cells had the bifid cell shape versus not. And this just shows on the y-axis the fraction of the cells that have the Y cell shape. And indeed, there's outliers. So the ones that are in the circles here are genes that are significantly different in their fraction of cells to take that morphology. And if you look at the functional categories of these genes that have increased bifid cell shape when mutated, it's multiple different functional categories. So it looks like there's multiple paths by which the bacterium takes this shape. Going into this study, it was thought that cell wall stress was the only determining factor to form that cell shape. And then another sort of outstanding question in the field is, why does it do this? Why does Bifidobacterium take that cell shape? And we kind of hypothesized going in that maybe it helps with survival in hosts, like mice or chickens that we had assayed. But it turns out that the mutants that have increased bifid cell shape tend to be sick in all of our conditions. They tend to be less fit than your average mutants, both in vitro assays and also in the host assays. So we don't know why Bifidobacterium forms that cell shape. All right, I'm gonna switch gears. We've gotten really good at generating these big data sets. If you can make, and genetics is amenable to your bacterium, we can generate this big gene phenotype map for a couple $1,000s in a matter of weeks. That's easy to do. But how do we propagate our insights to new genomes is not ideal. And the methods that we currently use to infer the functions of genes based on patterns of its phenotypes is done manually right now. And I don't want to be a scientist forever, so we need some, my wife's smiling, I definitely don't, like, we need some approaches to make this more automatable and done by machines, maybe. So the first challenging the propagation, this is more of a product of the fact that we can sequence so much, like I mentioned earlier. So we sequence so many new proteins. The other issue is that there's not much money for biocuration anymore. I don't biocurate, but it used to be there was lots of like model organism databases where people would like look through the literature and like, make the main insight from the paper and then put that into a database. That doesn't really exist anymore. Funding has gone down for that. So there's new approaches that are needed to propagate knowledge to new genomes. There's been a number of workshops that I've organized or I've been involved in. This is the most recent one that Valerie de Cressy-Lagarde at Florida organized. So there's a lot of talk around doing this, just not much action. And there needs to be better, communication between the data generators, people writing the algorithms, and the biocurators. But these communities are talking to each other a little bit, but we're still a ways away. Our approach is starting small. I like metabolic pathways because our discoveries, we can make discoveries on them based on the genetics data. So the tool that we are working on right now is something we call Gapmind. And we're taking a pretty conservative approach to manually curate different metabolic pathways. And specifically, we're doing amino acid biosynthetic pathways and pathways for catabolism of carbon sources. And so you can break down the pathway for making serine into 3 steps, SEER A, B, and C. And we define each of the steps. So for each of the steps, we identify all of the experimentally characterized proteins that we pull from these databases that are known to do that activity. We then supplement the experimentally characterized in the database genes with genes that we infer have that activity based on our genetics data. So this is another gene that we add to the database for characterized SEER A enzymes based on our findings from genomics. We do the exact same thing for all of these individual steps that are shown here for each of these pathways. What this allows us to do is take a new genome, in this case, Phytobacter inhibins, it's a marine heterotrophic bacteria, and we can put that into this database, and in 15 seconds, it'll tell us what's the most logical path that that organism uses to make each of the amino acids. And if each of the steps is highlighted in green, it means we identified a high confidence step for making that particular amino acid. Now, I like this example from Phytobacter inhibins because if you can see, it's all in green for methionine. It identifies a high confidence path for making methionine. And the reason why it finds a high confidence path is because the genes that I have highlighted here are ones that we added to the Gapmind database based on genetics. So if you're aware of the MET-H enzyme in E. coli, it's the vitamin B12 dependent methionine synthase. It's this big multi-domain protein. Phytobacter inhibins has split this one protein that's in E. coli into 5 separate polypeptides. Do the same activity. I also like this example because Phytobacter inhibins can absolutely make all 20 amino acids. And despite all the genetics that we've done and all the work that we've done, we have no idea how it makes threonine. You look at the genome, you cannot identify the enzyme that encodes THRB. Not there. No. There's still things to find in basic metabolism. All right. So the second big challenge is manually inferring the functions of genes is just not sustainable. I showed you this example previously of this poorly characterized gene that's involved in raffinose uptake. I'm sure you've read a lot about machine learning and the promise of machine learning to solve all of our ills of society and biology. I'm not a computational biologist. I don't write these algorithms. My simplistic view of this, you get all of this really high quality data, like genomes and genetics data, and you have all of this stuff. And you're going to train some machine that's going to give you the answer, right? It's going to give you more accurate annotations, will lead to more accurate pathway predictions, and that will lead to more accurate understanding of microbes and their interactions. I guess that's the dream. There has been a couple of papers recently on protein function prediction. And this is a pretty good illustrative example of like where the field is at right now. And so one's from this Google team led by Lucy Colwell. The other one's from Huimin Zhao at the University of Illinois. The math is different how they do their machine learning, but there's big similarities. They both use the Swiss Pro database for training their model. And both of these papers are predicting EC numbers, or predicting enzyme function. Is what they're trying to do. And in both of these papers, because you don't want to use the exact same data to train your model and to test it, they tested their model using enzymes that we had annotated using genetics data. So like a direct comparison of what us humans did versus what the machines can do right now. So how did they do? For the Google team, they did okay at like the more basic. If you know EC numbers, there's the 4 digits, and the first one is like the most basic annotation, like hydrolase. And then the subsequent digits become more and more specific. So you can see at the most basic level, they do pretty good, but at the highest level, they don't do particularly well. This green is how many correct predictions that they made. I think what's okay is they didn't even try making a call at the highest level in many cases, implying there's not enough data. For the other study, their algorithm is called CLEAN. They use a different thing, like a precision recall, to assess their performance. And they did pretty good, but it's not like substantially better than just BLAST P than naively saying that the best possible that you get by BLAST P is the function. It doesn't do so much better than that. And buried in the supplemental information from the science paper from the Zhao lab, I think gives some insight into what we need to do, which is this is showing the performance of their algorithm based on how many enzymes were in the training set in the Swiss Pro database. And so, once you get about 5 enzymes that are in the database, their model does like pretty good. But if there's only a couple enzymes, so 0 to 5 in their training set, their algorithm doesn't do very well. It makes sense, right? If the training set doesn't have examples, then you're less likely to be able to propagate that with machine learning. So my view is we need more data. These methods hold a lot of promise, but we need data, which is what we're trying to do. All right, switching gears again. This is one specific project that's been my longest funded project that I've worked on. And it is like very much in the Department of Energy space. This is Enigma. You can read the acronym here, but it's a big project with 100 researchers, multiple institutions, a number of PIs. We're trying to do mechanistic microbial ecology. I don't know what that means, and the PIs would all disagree exactly what that means, but that's how we sell it to DOE. We're trying to understand the impacts of the environment and microbes on each other. We study the Oak Ridge field site. So if you're aware of Oak Ridge National Laboratory and its history, you've seen the movie Oppenheimer. This is where all of the plutonium, uranium, I think, came from. Plutonium. Anyways, this is where a lot of the enrichment happens at Oak Ridge National Laboratory. Unfortunately, they didn't have the insight to properly dispose of the nasty materials they needed to enrich for uranium or plutonium. So they dumped it into these ponds. And so these ponds have lots of nitric acid waste. And of course, things kind of leaked. And so there's this big contaminant plume at Oak Ridge National Laboratory. This is our field site. Today, this is the old site from the 80s, I think this picture is from. Today it's a parking lot. Yeah, that's what it looks like today. There's been a ton of work at this site. It's a super fun site. DOE has spent a lot of money for cleanup. Tons and tons of money has gone into here. So a lot of measurements of the geochemistry and the microbiology have been done at the site by us and by others. The biggest microbiology insight, in my view, is that if you look at the most nasty of these wells that have been drilled to sample the groundwater, this Area 3 well, FW106, for example, is really nasty. It has like a pH of 0. It has high levels of uranium, high levels of nitrate, other heavy metals. Like it's a nasty environment. And if you do metagenomics, like what you see is Rhodanobacter. It's a game of proteobacterium. Rhodanobacter dominates the groundwater at these most nasty environments. So logically, like what's the basis? What's the genetic basis for its extreme phenotypes in this environment? So we've been able to isolate some bacteria from the site. This strain, FW10410B1, is one of our favorites because it's a genetically tractable one. And we've sequenced a genome of it and other isolates, and we get some insights about why it may be metal resistant. It has a higher number of metal efflux systems in it compared to your typical genome. So that's interesting. And some of these genes are acquired by horizontal gene transfer. That's all it needs. We made a transposon mutant library in this organism and did assays with different metals. And so we can say that this particular efflux system is important for cadmium, zinc, and uranium resistance. And this gene is important for other metals. It's all great, right? But it's really, really unsatisfying because if you know anything about all of these efflux systems, they're present in a lot of bacteria, not just Rhodanobacter. So to answer our question, like why are these bacteria surviving in this nasty environment? We don't have a satisfactory answer right now. Maybe like these are more active in Rhodanobacter, there's more copies. We don't know the answer. Even more unsatisfying is that the strains that we've isolated and done genetics on are not the exact strain that's the most abundant. So we've done lots of metagenomics, amplicons, metagenome assembled genomes. And the absolute most abundant amplicon sequence variant is a Rhodanobacter that we call top 1. It's top 1 because it's the most abundant organism across these contaminated wells. We have a complete genome. So before you ask, why can't you isolate? Having the genome doesn't help. It's not like some insight you can get by looking at the genome and say, oh, I can isolate it by growing it this way. We've never been able to isolate this bacteria. So it's very unfortunate. But we can get it into an enrichment culture. So Hans Carlson, who works in my group, developed a scheme to do like lots and lots of low volume enrichments. So we did around 36,000 enrichment cultures. We did 16S on all of them. And some of these enrichment cultures actually do enrich for that top 1 Rhodanobacter up to 30%. So I'm now of the mood, we may never ever isolate that exact bacteria, but we can get into an enrichment culture. And if we get into a stable enrichment culture, what kind of genetic tricks can we do to understand more about the biology of top 1? So I'll present really quickly 2 approaches that I'm really enthusiastic about that we're actively working on that are isolate independent. So they don't require us to isolate the bacteria to do these assays. We can run these assays off of the enrichment culture. The first is simply heterologous expression of DNA in another host. I don't need have an isolate bacteria for that. So this is work led by Yolanda Wong, who's a postdoc with Adam Arkin and myself. She now has her own lab at SUNY Buffalo. And as a proof of concept, we did this in gut bacteroides because it's easier to get a faculty job demonstrating a technique in human gut bacteria than in metal-resistant bacteria from Oak Ridge. But this approach is similar in essence to RB-TnSeq, except we're overexpressing genes instead of removing them with the loss of function transposon mutation. So we can take DNA from any source. It can be isolated DNA from isolate bacteria. It can be from an enrichment culture. It could be from low DNA samples, ancient DNA. It could be from anywhere. We shear the DNA and we clone it into an expression vector. We love barcoding everything, so our vectors have a little random DNA barcode sequence on them. And we're going to do long read sequencing to link the barcode sequence to the exact fragment that got cloned to that expression vector. So we generate a table. Barcode sequence is linked to this region in a particular genome. We can then introduce these plasmids to an expression host. In this study, we sheared up DNA from gut bacteria, and we use the sort of model gut bacteria, Bacteroides theta otomicron, as our expression host. you introduce these plasmids into the expression host, then you can compete the exact same ways we do for RB-TnSeq. You can compete all of these strains against each other. And what we're looking for is under some selective condition where the overexpression of one of these foreign genes leads to a fitness increase, is what we're looking for in these assays. These assays tend to have a little more stochastic effect to them. So we typically only focus on positive genes that have high fitness scores that we see across multiple replicate experiments. So this is one example, and I'll sort of walk you through these plots. So each of these little fragments, each of these lines represents a fragment of DNA from the genome of, in this case, Parabacteroides. These ones are from Bacteroides fragilis. And if you introduce each of these plasmids into B theta and grow the library on raffinose, these fragments have high fitness scores. So these are fragments of DNA covering this region that lead to an increase on raffinose when grown with B theta as the host. And so we were excited about this because this RAF-T1, RAF-T2, and RAF-T3, they're all poorly annotated TUN-B dependent receptors or transporters. And this final gene, this RAF-T3 from B fragilis, was a positive both on raffinose and on fructooligosaccharides. And these compounds have some similarity. They both have a terminal fructose group. And the thing I like the most about this is it sort of demonstrates the complementarity of doing overexpression assays, so gain of function assays, with our traditional loss of function assays. Because this RAF-T3 from B fragilis is 47% identical to the protein that I showed you earlier from Foveola vulgaris. So this is sort of like a geneticist dream, that if you remove the gene and you lose it, it's required for growth on raffinose. And if you overexpress it, it leads to increased growth on raffinose. So we were quite pleased by that. So I'm all in on this technology, right? I think this is great because they can allow us to assay DNA from environmental sources. Doesn't matter where the DNA comes from. It does require us to do DNA cloning a little better. So using less input DNA. So for the folks who study ancient DNA and like how you clone low amounts of DNA into an expression vector is technically a challenging problem. We have a preprint coming out soon on doing genetic complementation at scale. So you can make these overexpression libraries and introduce them not into a wild type strain, but introduce them into a knockout strain. And ask for that gene that's knocked out, what are all the fragments and genes encoded on them that can rescue that activity? I think that holds a lot of promise. And we're currently trying this on our top one Rhodanobacter enrichment right now. So again, we can't isolate that bacteria, but I can totally get DNA from it, from the enrichment culture, and overexpress it. So the final thing is the second isolate independent approach is this microbial community editing. And this is all work led by like three at the time really great postdocs, Ben Rubin, Spencer Diamond, and Brady Kress, who were working at the Innovative Genomics Institute with Joel Banfield and Jennifer Dowda. And this is, I think, early stages, but it's really, really exciting. So they developed this two-step process for community editing, and it's kind of illustrated in this plot down below. In the first, that given a community of organisms, you first want to know which members of that community are accessible by genetics. And so Ben developed this approach called environmental transformation sequencing, or ETC. You deliver an untargeted transposon via different delivery methods, and you can do sequencing to figure out which community members acquired an insertion. And so you figured out that, the blue organism definitely looks good, and electrobolt is probably electroporation is the best way of doing DNA delivery. Once you know which members of a community are accessible by genetics, you can then do a targeted edit. And it takes advantage of CRISPR-associated transposons. Guide RNA transposon allows you to deliver a transposon to a specific location in one genome. So this example is a guide RNA against a blue organism with some cargo, and we can deliver that just into one spot of the genome of the blue bacterium. We weren't the ones who identified these CRISPR-associated transposons. We leveraged them for our community editing purposes. We tested a few different systems. The one from Vibrio cholera, or VC, DART, turned out to be the best one. DART, again, just an acronym. People call these CRISPR-associated transposons now. And it had a great on-target effect, right? Rarely inserted into the wrong spot. And it all looked fantastic. And so we had two sort of proof of concept applications. The first is targeted enrichment of an organism. So given a community member, we can design a guide RNA with its CRISPR-associated transposon, just the one genome. So this is our starting organism here, our starting population of organisms, and we deliver a guide RNA just to the blue bacteria. And in the transposon cargo, we can put anything that we want. In this case, we put an antibiotic resistance gene. And so we can grow on the antibiotic and select just for our Klebsiella michiganensis. We can repeat the experiment, this time having a guide RNA against the maroon bacteria. So Pseudomonas simiae. And likewise, we can just enrich for that organism by delivering a transposon to that microbe. Of course, longer term, you don't want to introduce antibiotic resistance to your community. So we also had one more proof of concept where we delivered a carbon utilization pathway. So none of these bugs could natively use lactose. So we put the lactose utilization pathway into the transposon, delivered that specifically to Pseudomonas simiae, did the enrichment on lactose, and we enriched for that community member. This is early stages. But this is going to go fast. Like the IGI Institute at Berkeley got a donation for 70 million. I'm not part of this, but they got a donation for $70 million to pursue this line of work for applications in, I think, like methane reduction in cows and for human health applications. So I expect this to move pretty quickly. There's a lot of technology things that need to happen. We're trying to get these into phages now because phage is probably the best way of delivering your cargo to most members of a given target organism. We're trying this now in our top 1 Rhodanobacter strain. We're trying to figure out how like realistic environments would influence your ability to do this editing. This is like a shameless plug for other things we're doing on our project, but with this MCAFE's project, Trent Northern's lab has developed these EcoFab devices for doing controlled plant microbe interaction studies. And these things can be automated. So we have a big robot that can run through an image and do assays for like hundreds of these in an automated format. So if you're interested, feel free to reach out. With that, I'd like to thank individuals as part of Enigma who did most of the work. And so Morgan did the vast majority of the data mining to make new annotations. And Ruby's lab did most of the isolations, and Yolanda developed the overexpression approach. And it was the folks highlighted here who developed that microbial community editing technology as part of MCAFE's. And with that, thank you for your attention.
Thank you for your talk. We have questions from trainees.
Yes. Thank you so much for all those wonderful stories and methods and interesting things. I'm really curious for the RB-TnSeq. When it's completed, it seems like you've done it on multiple Pseudomonas fluorescens strains or something. When you do it on very similar strains but with different backgrounds, how different are the results for the fitness of certain?
Yeah, that's a good question. So the question is, we've done RB-TnSeq, for example, on multiple related Pseudomonas fluorescens strains and how similar the fitness results. We've only looked at it closely for metabolic enzymes. So genes involved in catabolism of like nitrogen and carbon sources. And it's pretty similar. We have projects now that try to do genetic interactions at scale. So the look for cases where an organism may have redundancy. So one Pseudomonas fluorescens may only have one isozyme for one activity that has, another one has more than one. We haven't seen that much. But we haven't done something really systematic to look at like phenotypes for like accessory genome genes and things. There could be something interesting there to do. And if you were to do it, I think it sort of would exist in microbiology right now. The biggest data set for gene fitness data for relatives would probably be a Pseudomonas fluorescens subgroup.
Thank you.
Any more questions?
Yes.
Have you tried the RB-TnSeq on any fastidious bacteria? Then what I have in mind here is obligate intracellular bacteria that are really hard to genetically work with. So we would benefit from a technique like this if it was possible.
Has anybody ever genetically modified one of those?
I imagine people have tried CRISPR and have had infrequent success, but that your question makes me think that there would be some challenges. And
I frequently like tell collaborators that like my approaches don't allow you to get genetics working in your host, like to get over that initial barrier. If genetics is accessible, then our approaches allow you to kind of go, like kind of crazy on like the sort of scale of assays you can run with the organism. But that sounds hard.
right.
That sounds really good.
But I leave that to you because you're clearly the expert.
Maybe I often like say when I give talks like I definitely focus on the easier organisms to grow and cultivate. So there's a lot to learn. I think even
I
for
would
the
too.
easier organisms. But I do think I'll leave it to like the students here to maybe take some of the things that we've started and you can work on intracellular obligate symbionts of insects and other things that are more challenging. I'm after the lower hanging fruit. But we have like I said, our Archaea works now. We're getting better with gram-positives. A number of labs have done similar approaches in fungi and non-model yeast strains as well. So we have this working in Trichoderma atraviridae. So Trichoderma is an important fungi. So it can work in other systems as well.
Thanks.
Yes, I'm just curious about your system. Since you're using machine learning, could it be possible to, for example, if you want to analyze the contamination in a lake, to with your system know about the contaminations if there are, the kind of metals and bacteria at the same time, extracting just just once the water, the sample water?
Oh, yeah, I don't know a ton about that. Like the example that we played with, the machine learning example I gave here was basically would give you no insight at all about the ecology of the organisms from which those proteins were derived. It's just based on protein sequences and trying to predict an enzyme commission number out to as many digits as you possibly could. I know there's approaches that people are thinking about as like classifiers for like microbiome studies, but I'm not an environmental microbiologist. So like I know it's like a microbiomist. I don't do much like microbiome stuff. I have my projects, like we definitely do microbiome stuff, but my lane is not doing very much of the microbiome work. I think the lesson that we have from machine learning is that like, yeah, it's a little bit like sensationalized right now. And at least for the stuff that I work on, like protein function discovery, it's not quite there. Like the hype doesn't match the reality.
Thank you.
So you were mentioning with your overexpression approach and needing a fair amount of genetic material to actually clone to get that to work. And I'm wondering, is the DNA synthesis getting cheap enough, or do you see that horizon where at some point you don't actually have to clone based on what's in the environment? You can just tells, Sigma or some other
Yeah, I was
company
talking to
synthesize
Laura
this.
on the way over about this. I do think it's getting closer, but it's still prohibitively expensive.
Yeah,
I think if you had like a really targeted hypothesis about like one system or one operon, you could do it, but DNA synthesis, if you wanted to synthesize, say systematically, say you had a metagenome assembled genome complete from some organisms, and you wanted to systematically overexpress all of its genes, it would cost you a lot of money. Like a synthesis is cheap for a handful of constructs, but expensive for doing it. But maybe, I think that there's companies, obviously, that are trying to make synthesis cheaper and better and stitch together bigger pieces of DNA. Just not quite there. Like DNA sequencing costs have gone down so much more. I think synthesis has gone down, but not quite at the same rate. But maybe in a couple of years, absolutely. I would probably synthesize everything rather than shear it at that point, because if you put, when you synthesize it, you could put, for example, the right ribosome binding site for your host organism on there. There'd be other things you could do to make it better.
Right. And we're just thinking about how we have really poor data, right, for these machine learning algorithms, right? We need to improve that data. Do you think it's possible to map out if we had X number of taxa across X number of phyla, we had 5 representatives from each of those, right? That we could get this sort of baseline data that we can then use to feed into these databases to, get everything we
That's a
need.
great question. And we've never done a systematic analysis of that, but like naively, it's definitely the case that we get more bang for our buck because there's different protein families and the homologs are more distant to do, new bacteria that we haven't done in the past. So things that aren't Pseudomonas fluorescens. Kind of bemoan the fact that collaborators often want to do Pseudomonads because they'll work and they're interesting. Just from like my point of view, it's not quite as interesting from a discovery point of view as if you got, an intracellular symbiotic working. Like, it'd be more exciting. So we're kind of approaching that. So for a lot of our work, we end up doing isolates that we get from our projects that we do genetics on. But I'll supplement that with type strains that I buy from stock centers as well. Sometimes to fill in missing gaps where we haven't, studied, some particular phyla or genera.
Thank you. Let's take our.